dear cutie pies, how are you all? How's the Josh? I hope everyone who is watching this video is doing absolutely crazy good. Okay, now one very important thing. Here I am with the purpose that is to solve a question paper. Well, where did I get the question paper from? Well, this was given for the preparatory exam of biology, PU2 preparatory exam, on 21st of January 2025, that is yesterday. So we thought we will bring out the solution for this video, this question paper, and it was given for Bangalore North category. Okay, so what is the use of this? You will get to know what type of questions have come. Okay, you will get to know whether whatever we have taught you, is it coming or not? Okay, in the Brahmastra series, it is actually useful or not, and things like that. And you will also see any new questions which have popped up. So according to the little analysis that I have done throughout, I think all the questions are from Brahmastra series itself. So that is a good news from us. But you have to do your analysis also. Okay, it's not just me telling you that whatever I taught has come. It should be like you also should be very sure that whatever I have taught have come. For that, you have to be with me and check. Okay, so let's get started. Section A consists of 1 marker question, my dear children. So always, as I said, in biology you cannot expect straight questions. For example, questions would not be repeated as such. Okay, the concept will be repeated. So lactational amenorrhea, the first question that I am going to show you over here, about this I have taught you. Now you have to use your brain and think that what type of questions would be coming. So lactational amenorrhea is something that we have completed, but they have reframed the question. Now let's see how. Okay, so statement 1 is that lactational amenorrhea is the absence of menstruation during intense lactation. That is, when the mother is breastfeeding at this point of time, a mother would not be menstruating. That is, no ovulation would be happening, no release of eggs will be happening. Okay, so if there is no release of eggs, obviously sperm cannot come and fuse and no fertilization can happen. Okay, so which is why lactational amenorrhea means during lactation there is no menstruation. Now that statement is correct, but statement number 2 says this is highly reliable method of contraception. So I'll tell you, this is a good method of contraception for sure. Okay, because egg is not released, sperm won't be coming and fertilizing it, that is for sure. But is it highly reliable? No, this is not highly reliable method of contraception, which is clearly mentioned in your NCERT textbook. Definitely it is a contraceptive method, but not a highly reliable contraceptive method. So I'll say statement 1 is correct, statement 2 is what? Incorrect. Okay, so that is the answer. Answer number C is the answer. Okay, now the next question that we have. The first amphibians that live both on land and water evolved from, again something that we have already discussed all the times, the questions comes from lobfins and shrews, and I've always told you, the one that came between fishes, okay, the fishes, they evolved into amphibians, okay, fishes were in water, amphibians are the one that lives in both water as well as land, and then came reptiles, then came birds and mammals, everything, okay. So from fishes to amphibians, in this transition phase, who was that person, or let's say that organism which came in between? Okay, which lived, which was considered to be evolving, and which was considered to be living on both land as well as water. It was lobfins, my dear children. Okay, so this is a direct question again from your NCERT. So what is the answer for this? I am not writing the answer. Let's say answer number C is the answer. Okay, now if the question was what is the connecting link between reptiles as well as mammals, it would have been shrews. Okay, cool. The site where lymphocytes interact with antigens and proliferate to become effector cells are. So it is directly asking you, where does the lymphocyte go and do its actual work? What is its actual work to fight against diseases? Now always remember, the primary lymphoid organs are where the lymphocytes are made, right? That is bone marrow and thymus. Bone marrow and thymus is where lymphocytes and all the things are being made and they are matured. But they do not do their action there, they do not do distribution there. They go to secondary lymphoid organs such as tonsils, such as Peyer's patch, such as spleen, such as lymph nodes and things like that. Again, we have discussed in the 5-marker question the same thing. What are the primary lymphoid organs, what are secondary lymphoid organs and things like that. So I told you, you get so many questions, my dear children, so you should know the concept, otherwise you are going to get it wrong. So the site where lymphocytes interact with antigens and proliferate to become effector cells are, where do they show their effect? In the secondary lymphoid organs. Can it be thymus? No. Lymphoid node is correct. Lymph node is correct. This is not correct because this is a primary lymphoid organ. Bone marrow again primary, thymus again primary. Peyer's patch and tonsils. Now this is something that we needed. Both of them, they are secondary lymphoid organs. Again bone marrow is this, which is why this will be wrong. Spleen is correct, but this is wrong. Cool. So C becomes your answer. Next question. Come on. Yeah. In the given diagram PBR 322, identify the restriction sites A, B, C and D. Chalo, let's start. So always remember, one patch over here, another patch over here. When they are opposing to each other, no, this and this, one will be ampicillin resistant gene and the other one will be tetracycline resistant gene. In between them you get two things. One is origin of replication and the other one is, this one will be ROP. This is a protein needed for replication to happen. Okay. Now over here you have three sites for other what do you call them? Restriction enzymes. Okay. And this one D1 is what you call as HIND3. HIND3. So A, if it is ORI, so just look for A. Over here there is only one option with A as ORI. B will be what? Ampicillin resistant, C will be tetracycline resistant and D will be HIND3. So this becomes your answer. So look for ORI, it becomes lot more easy. So when there are two in between other two, the smaller twos between longer twos, that means this is ORI. Cool? Yes? So this becomes your answer. Next question. Yes. Identify the incorrect statement. The transgenic cow Rosie produced milk enriched with alpha-lactalbumin. Yes, that is true. She produced a milk that is rich with human protein, that is alpha-lactalbumin. Now, Bt toxin genes Cry1AC controls corn borer. So, Bt toxin, I hope you know that Bt Bacillus thuringiensis is a bacteria which releases which has certain genes in them, such as Cry genes. Okay, and one such Cry gene is Cry1AC. Now, Cry1AC gene will be producing some inactive toxin, and this inactive toxin, when it is taken up by some insects, okay, in the gut of the insect, this inactive toxin becomes what? Active, and it is going to kill the insect. So it is asking which is the insect that is getting affected with the help of Cry1AC. So it is not corn borer, my dear children. Always remember, Cry1AC and Cry2AB is for cotton bollworm. Cotton bollworm. How do I remember it? 1C. First letter starts with C. Second letter ends with B. Cotton bollworms. Now you might say corn borer is also the same thing, ma'am. So for corn borers we have one thing. Cry2AB. Okay, Cry2AB. So C and B. Corn borers. Sorry, not 2AB, 1AB. 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 Here 1C, second word starts with B. Again, it's kind of confusing, but yes. So this is the wrong thing. This is a wrong thing because here it is corn borer, it is not corn borer, it is cotton bollworm. Now human protein alpha-1 antitrypsin is used to treat emphysema. That is correct. The commercial production of human insulin was possible by transgenic species of E. coli. That is also correct. Everything we have studied already. Okay, now you might ask, why is ma'am going very, very fast? Because my dear children, we just have to discuss the paper. We do not have to, think through it. Everything, each and everything that I am mentioning over here, we have already studied in Brahmastra series. Go watch the series. It will be really, really helpful for you. I am not telling you for views or likes or comments. I don't want anything. Do not like the video. Do not comment the video. Go watch it. It will be really useful. Put it in 2x speed. Watch it. Okay, it will be really helpful. Now, chalo, identify the correct sequence of steps in the process of decomposition. Okay, decomposition as in breakdown of the content which is there. Okay, which has to be degraded. Okay, now over here the first step always will be the detritivores such as earthworms and things like that. They eat the content. They eat the content and they poop something out. So basically they are eating complex thing and they are going to poop out what? Simpler things, which is very easy to be broken down now. So fragmentation is the first step and that is done by detritivores. Okay. Now after fragmentation is something that is known as leaching. What happens? Water soluble content, okay, water soluble content which is there, it will slowly seep into the soil and will stay there. It will form rock or things like that. That is what you mean by leaching. Now whatever content is there, the organic matter is still left behind. After fragmentation, you get what? You breaking down complex thing into simpler things, but they're still there, right? Now you have to break down the organic content which is there into inorganic things, and this is known as catabolism. Okay, after that, what happens? While this breakdown is happening, this is not a one-step process. It takes a longer time. So in the middle, from complex organic substances to simple inorganic substances, you reach a phase which is known as, there is a formation of something that is known as humus, which is not totally broken down phase. It's still there. The organic content is still there. It has to be broken down. So this phase of formation of humus is known as humification. Later on things are later broken down, all the minerals and everything is been taken out. This is known as mineralization. Okay, so this is the flow. Please remember this. So this is the correct sequence of the steps that are involved in the process of decomposition. Okay, now yes, answer is A. Okay, why is it not working? Work. No. Chalo. Chanda and Bastar areas known for secret groups are found in. What are secret groups, my dear children? For example, people, they are conserving one area. Why? Because it is helping in their religious beliefs and things like that. It is supporting their religious beliefs, which is why they are conserving a particular area. When they are doing so, what is happening? All the other things which are there, over there, they are also getting conserved. For example, animal groups or plants, whatever is there, it is also getting conserved. So, which is why you call it sacred groves. Now, sacred groves are found in lots of the other places, okay. Now, you have Chanda and Bastar areas. It is found in Madhya Pradesh. It is found in Madhya Pradesh. And I profoundly remember, I taught you this, that you have to remember the different names of secret groups. So I have brought you here also. So Khasi and Jaintia Hill is in Meghalaya, Aravalli Hills is there in Rajasthan, Western Ghat regions of Karnataka, Maharashtra, Western Ghat regions of Karnataka and Maharashtra, and Sarguja, Chanda, Bastar areas of Madhya Pradesh. In Meghalaya, the sacred groves are the last refuges for the large number of rare and threatened species. Now, on the basis of this particular statement from your NCERT, also questions have come. Okay, which I have discussed. Meghalaya is what? In Meghalaya, there are sacred groves are the last refuges in the way that they can protect a lot of things. Okay, so please remember all these small details. So your answer becomes what? Madhya Pradesh. Yes, your answer becomes Madhya Pradesh. Yeah, it is given. Yeah, sorry. So from the given statement below, choose the option that is true for a typical female gametophyte of a flowering plant. Female gametophyte, as in you're talking about what, my dear children? You're talking about, tell me. You're talking about the embryo sac, right? Now, what is true for embryo sac? What is true for embryo sac? How many nucleus does it have? How many cells does it have? Now, let's see it is working or not. Anyways, so over here, if I make an embryo sac, this is egg cell. These two are synergy cells. These three are antipodal cells, and then you have nucleus. So over here 1, 2, 3 nucleus, 1, 2, 3 nucleus and 2 nucleus extra, which does not have cells surrounding it. So what do you call them? You call it 8 nucleated condition, right? How many nucleus are there? 8 nucleus is there. What about the cells? If we see 1, 2, 3, 4, 5, 6. There are 6 cells, and these 2 nucleus are in the central cell, which starts from here and goes up till here. This whole thing that you see over here, this is a central cell. So if you talk about the cells, how many cells are there? 1, 2, 3, 4, 5, 6, and then this whole thing is what? 7. So this is correct. This is correct. The female gametophyte, that is the embryo sac, has how many nucleus? 8 nucleus and 7. So this is correct. What you have to find out? You have to choose what is true, right? Now it is free nuclear during the development. What do you mean by free nuclear? As in the nucleus keeps on dividing, but the cytoplasm is not covering it. Is it true or false? Yes, it is absolutely true. It is free nuclear during the development. Slowly cytoplasm comes around it, and the cell wall comes around it, and everything happens, and they are divided into different cells. So this is also true. So I say A and B is true. Anyway, 2 options we have to find out. The first 2 options are correct. Okay, A and B, let's see. Is the C which says that A and B options is correct. And with this, we get C answer. Cool? Chalo. Which of the following pairs correctly matches the disease and pathogens causing it? We have to find the correct match. I always tell you, whenever you have to do some correct thing, what do? You have to underline, because we start searching for incorrect things. Don't do that. Okay, we do not have the time. So over here, we have to find the correct match. Typhoid, salmonella typhi. First option itself is correct. Typhoid is a bacterial disease which is caused by a very famous bacteria that is known as salmonella typhi. What is the test for it? If you know, comment it down. What is the test for typhoid? There are only 2 diseases given in your NCERT who has test. Okay, so you have to tell me the 2 diseases name. One is typhoid and the name of the test, and the other disease is viral disease. You have to name that disease also in the comment section if you know right now. So what do? Write 9 and then tell me the answer, otherwise I will also forget. Okay, now pneumonia, Haemophilus influenzae. Malaria is caused by Plasmodium. Ringworm is caused by Trichophyton or Epidermophyton and things like that, but not all this thing. Okay, Ascaris lumbricoides causes Ascariasis, Antamoeba histolytica causes Amoebiasis, Haemophilus pneumonia is not there. So yes, so answer is A. Now, when is it possible for a woman to be color blind? When is it possible for a woman to be color blind, my dear children? Color blindness is a X-linked recessive trait. It will be present if both are like this. Well, that was a fun thing. Okay. I'm not as talented as you children, okay? Okay, did something go? Yes, something went. Okay, anyways. So, my dear children, remember, color blindness will be only shown when two of the alleles are affected. It is a recessive trait and it is an X-linked recessive trait. So, when would be a woman colorblind? You should be receiving one X from the father. You should be receiving one X from the father and the other X from the mother. Then only you will be having color blindness. Okay. Now, over here, if one X from the father is like this, don't you think the father is colorblind? Because there is no other X to over-dominate it. So, anyway, this particular person should be colorblind. Now, what about the mother? What about the mother? Mother can be like this, okay, because one X, any of the X can come, or she can be like this also, where she is just a carrier. Now, suppose this X comes and from here also X comes. So, the person becomes colorblind. So, the father has normal vision, the mother is a carrier. No, the father cannot have normal vision. The father is colorblind, the mother is normal vision. She cannot have, okay. The father is colorblind and the mother is normal vision. She does not have normal. She is a carrier. That will be more correct. Okay. So, the father is colorblind and the mother is a carrier. This becomes totally correct answer. Okay. This is the total correct answer. So, D is the correct answer for this. Okay. Now, the shape of the age pyramid reflects the population growth status. In this context, the given age pyramid indicates that the population is what? How can you understand? Look, here this is the pre-reproductive organism, as in they are not in the reproducible age, but soon they will be in the reproducible age. So, pre-reproductive organisms are this much, reproductive organisms are this much, post-reproductive are this much. So, whenever you see pre-reproductive, okay, when they are less, that means it is going to be declining. Okay, and the others are more, but they are less. That means it is going to be declining. Why? Because these are the organisms which has to reproduce, and if their number is less, then obviously, do you think the population will grow? The population would not be growing, my dear children. So, I will say it is declining. I'll say it is declining. Okay, so answer is declining. Again, this is also something that we have already covered. Now, choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. So, over here zona pellucida is given and it is surrounded, okay. So, this is the egg and it is surrounded by zona pellucida, okay. Listen to me very carefully. Zona pellucida is something that is made from the secondary oocyte, my dear children. Who makes it? Secondary oocyte. Primary oocyte divides into what? Secondary oocyte. I hope you know that. Secondary oocyte is the one that gives rise to an extra layer that is known as zona pellucida. Now, zona pellucida which is there, it is what say broken down. When is it broken down? After fertilization, not before fertilization. After fertilization, this particular zona pellucida that you see, okay, when they come and fuse, my dear children, actually it is kind of incorrect to say that, but I don't see any other options as well which is much better. Okay, if I say before fertilization, then it can be any time before fertilization, right? So, obviously, I would have to say after fertilization, when the sperm comes and meets with the ovum, okay, at this particular point of time, that is secondary oocyte, at this particular point of time, what happens? The zona pellucida and the receptors on the zona pellucida and the proteins on the egg will come and bind to each other, and the sperm will be releasing lots of enzymes, sperm lysins. This will break open the zona pellucida and things like that. So, I will say during fertilization, but since there is no option, I will say after fertilization, this becomes more correct. Okay, so your answer becomes D. Now, match the following: Alec Jeffrey dash, F. Sanger dash, Jacob Maynard dash, Griffith. So, everyone, I think that you know about Griffith. So, I always start with Griffith. So, Griffith did the transforming principle experiment that he did with the mice, my dear children. So, Griffith with mice. D with 2. D with 2, there is only one option where D is with 2. Let's see the other things. A with 4. Alec Jeffrey was the one who gave down DNA fingerprinting. Okay. Now, B with 3. F. Sanger, Frederick Sanger was the person who annotated DNA sequencing. Okay. Now, Jacob and Maynard, this is again something famous, they gave the lac operon. So, this answer becomes the correct answer. Please remember the names of all these people, really important. So, these type of questions you never expect to come, but yeah, they come. Okay. Yes, they come. So, you have to remember a lot of things to get to know this answer. You get it? But at least if you get a clue for this, for example, Griffith. Okay. Sometimes they do not give this much easy options, but yes. So, if you know Griffith, everything becomes easy. So, anyhow, you have to study all the name of the people who are behind this thing. Okay. Now, arrange the following in the order of evolution. We did so many times. So, we have okay, Australopithecus, Neanderthal man, Homo Homo habilis, Homo erectus. My dear children, Ramapithecus over here. First, Ramapithecus were like human-like organisms, okay, human-like organisms. From Ramapithecus, you got Australopithecus. So I'll say C comes first, okay. After C comes B. After B, Australopithecus, basically you come Homo habilis, Homo habibi, the first human-like organisms over there. Okay, Ramapithecus had human-like feature but not exactly. From them, who came? Australopithecus came. From them, Homo habibis came. Okay, Homo habilis came. From Homo habilis, you got what, my dear children? Homo erectus, E. And then Neanderthal man. And then Neanderthal man. So this becomes the correct answer. Okay, I always remember with Homo habilis, it becomes the easiest thing. So habibi, we human beings say we are habibis, my dear children. Okay, so basically, the human-like character came from Homo habilis and then came Homo erectus because they will be able to stand erect and things like that. Okay, so this becomes the answer. Chalo. Next question. Identify the correct statement. HIND2 cuts the DNA at specific sequence of 6 base pairs. HIND2 was the first restriction enzyme to be found and it is the one that is actually going to cut at 6 base pairs. The recognition site for this particular enzyme, restriction enzyme, is having 6 base pairs. This is given in NCERT. This is correct. I have told it 10,000 times, so this is the correct answer. Microinjection is a technique of introduction of our recombinant DNA into the plant cell. No, it is microinjection is done in animal cell. Biolistic is a technique of introduction of recombinant DNA into animal cell. No, it is for plant cell. Okay, retrovirus is commonly used for cloning vectors in plants. It is commonly used vectors in animals, not in plants. Again, this is wrong. This is the only one that is correct. Cool. Chalo, next. Now we are come with a fill in the blanks question. Again, something very easy. The interstitial space in seminiferous tubules consists of immunologically what? So in between the seminiferous tubules, you know, there's a testicular lobule. Okay, listen. In the testis, there are so many testicular lobules. How many? 250 of them. Inside each testicular lobule, there are 1 to 3 seminiferous tubules. So I'll make box. I'll make dabba. Inside that, too many dabbas. So these are the seminiferous tubules. This one, this one. So this space that you see over here, that is known as interstitial space, which has Leydig cells, my dear children. What cells does it have? Leydig cells, which is responsible for the production of hormones, male hormones. But apart from that, they also have certain immunological cells, the ones that will fight against all the diseases. So I will say immunologically competent cells. So this becomes the answer for the 16th question. Okay, now dash is the most feared property of malignant tumor. What do you mean by malignant tumor? Malignancy. That means the cells which are there in the tumor, it can roam around anywhere in the body. Okay, that is why we say the cancer has spread. Okay, in malignant tumor only, the cells which are there, which are participating, they spread around throughout the body and cause cancer. Okay, so they are showing something which is known as metastasis. Okay, metastasis. So this becomes the answer. So for 17, this is the answer. For 16, this is the answer. Okay, now dash is a white, odorless, bitter crystalline compound which acts as a depressant. White, odorless, bitter crystalline compound. It is opioid, my dear children. It is some opioid. What is it? Heroin. And heroin is known as smack. Okay, it is known as what? Smack. It is a depressant. It will make you go into depression. Okay, so what is it? It is smack. Okay, now many members of genus dash form mycorrhiza. Glomus. Okay, fungus, genus of fungus, that is known as glomus. Now GAATTC is the restriction site for 1, 2, 3, 4, 5, 6. 6 base pairs are here also. But this is a restriction site not for HIND2 but EcoR1, very famous enzyme. What do you have? EcoR1. Please remember this restriction site. Everything I have taught. Okay, nothing out. Fine? Chalo. Next thing. Section B consists of 2 markers, my dear children. Differentiate between albuminous and non-albuminous seeds. So over here you have to mention these points, that is albuminous seeds have persistent endosperm, my dear children, which will be that stores food for the developing embryo. Okay, there will be some amount of endosperm still left back. Okay, and they will be just having one cotyledon. But anyways, example, what maize and castor are non-albuminous. They have little or no endosperm at maturity, as it is absorbed by developing embryo for the formation of 2 cotyledons. 2 cotyledons, which is why you call them dicots, and they are known as monocot. Okay, example is pea and bean. Over here, maize and castor. So these points you have to write. If you're not writing, you're not getting marks also. So when I discussed albuminous and non-albuminous, I didn't think that there will be a 2-mark question arising from it, even though I explained. So these type of questions definitely arise. So you need to know how to frame your answer, write in points, underline what is important. So I have marked it in, what's a bold, but you should be knowing how to write in points. Okay, chalo. Write the reasons for infertility in humans. Again, this is something that is twisted and given. Okay, this is twisted and given. This is there in your anxiety, very easy thing, but still, you know, sometimes when we are in that awe of exam, we do not remember every single thing, or we do not know to frame what we are thinking of. So this is for them. Okay, I hope the answer will make you understand how you need to write. Okay, now listen to me. Infertility in human beings, it can be from male or it can be from female. In the case of males, mostly it is there is loss of motility of sperm. The sperm are not moving very much, or probably in the semen the sperm count is way too less. Okay, the sperm count itself is way too less, and you should be knowing that there should be so many sperms required for fertilization, because only one guy will go and fertilize. Just all of them, they're just going to die off there, but still you need a lot of them. Only the fittest one will be going and fertilizing the ovum. So low sperm count, or basically what do you say, less motility of sperm, is a big reason of infertility in the case of men. What about females? In the case of females, probably again there might be some diseases, okay, in them, for example PCOS or things like that. Anyways, where the ovary is not producing egg regularly, the ovary is not producing egg regularly, or probably suppose fertilization has happened, still the uterus which is there, it is highly unsuitable for implantation to happen. If implantation is not happening, do you think the embryo will develop into a fetus, my dear children? No. Again, this is a reason for what? Infertility. Okay, so low sperm count or poor sperm quality in males, a low sperm count or poor sperm motility can reduce the chances of successful fertilization of the egg. Uterus is unsuitable for implantation, and irregular absence of what? Ovulation, often caused by hormonal imbalances, is the reason for females. So you should be writing in 1 and 2 points properly, otherwise you are going to lose your marks. Cool? Chalo. Next, in Drosophila melanogaster, the percentage of recombination is very low in cross between yellow-bodied and white-eyed with its wild type. So whenever it is mentioned that the percentage of recombination is very low, that means just understand there are two friends and they are not allowing anyone else to come in their gang because they are too close. So just understand that these genes, okay, probably the genes which are there, they are closely linked, my dear children. The genes, okay, both of them, they are closely linked. Now, if I try to separate them, okay, if I want recombination to happen, what do you mean by recombination? Suppose this is another one, another chromosome, and here this part goes, here this part goes. Now, I will get a new chromosome which looks like this, okay, where this will be there, and on the other hand, these two genes are still there together. Why? Because they were closely linked, and these closely linked genes, that means there is less amount of recombination. They are not allowing new combination to form because they are very close to each other. So the percentage is very low in the cross between this. Why? Because the genes are closely linked. Now the percentage of recombination is high in a cross between the white-eyed miniature winged with its wild type. Why is it way too much? Probably because one gene is over here, another gene is over here. Okay, the one gene is over here and the other gene is over here, which is why recombination is more. I'll explain how. So this part of the gene went there and the other part came here. Now you got a new recombination. Okay, this and this is a new combination that you're having. You didn't find it over here. You get it? Why is it happening? Because these two genes are not closely linked. They are loosely linked. What, children? They are loosely linked, which is why recombination, new combination of chromosomes and everything, genetic material can be seen. So percentage of recombination is high in a cross between this. Why, my dear children? Because, why? Because they are loosely linked. So how do you frame the answer? Yellow body and white eye are linked genes on the same chromosome. The low percentage of recombination suggests that these two genes are closely linked. Okay, this one. Whereas if you have high percentage of recombination, that means it is loosely linked. They are farther placed apart. Okay, they are very, very apart from each other. That is what you have to write. Write in points and you're going to score really good marks. Please understand, underline what is mentioned over here. Again, this question also, in some way we have discussed, okay, the concept we have discussed. Now they have put it into different question format and given to you. Now if you are aware of the concept, won't you be able to know the answer as well? Yes. So which is why I am telling you, watch the video. Now what are UTRs? Where are they located on an mRNA? Listen to me, UTR means untranslated regions. Okay, so usually mRNA is translated to what, my dear children? It is translated to proteins, right? Now in front of the mRNA strand, okay, in the 5' end, in the 5 dash end area, in the 3 dash end area, you have untranslated regions. Okay, untranslated regions which are very, very important for translation process to happen successfully. Okay, so at the 5 UTR, as in the 5 dash end, there is UTR, which you call as 5 dash untranslated region, located upstream of the coding sequence, as in from this point where the coding starts, as in when the translation starts, just in front of it you have this particular thing. It plays a major role in regulation of translation initiation and stability of the mRNA. So it is the thing that will be helping in the process of translation to be initiated and also for the stability of the mRNA. Now the 3 dash end also you have untranslated region. After what? After the stop codons. After the stop codon, okay, in translation unit, I hope you know that there is a start codon and there is a stop codon. In the middle there are areas which getting translated. Now at the end, after the stop codon, you have untranslated regions which is responsible for, again, for mRNA stability, localization and translational efficiency. So these are the things that you have to write down. Now what do you need to importantly write down, even if you do not remember everything, is that it is present upstream and this is present downstream, and this is very important for what? Translational initiation and the stability of mRNA. Even if you write these many points and just exclude this point, it is also fine, but you have to write it. Again, something that question that comes again and again and again. So please remember, this is a very small line in your NCERT but gives you a question every time. Okay. Now next question, what is divergent evolution? Give an example. Divergent evolution, my dear children, always remember, divergent as in two organisms are going in two different directions, divergent. Okay, so they were having a common ancestor. They were having a common ancestor, but because of what say habitat requirements, okay, but because since they went into two different habitat, they have evolved differently. Okay, for example, your siblings, your brother or sister might have gone abroad for studies or to some other state. Now you are initially same, okay, you are having same food, same things, but now you are in two different areas, okay, and now you have evolved differently. This is what you mean by divergent evolution, where you have a common ancestor and from there you are moving and now you have different character. Okay, so the internal structure, everything will be same. Internally everything will be same, you and your brother, everything, you are thinking everything might be same, but when you go around there, the dressing and everything might be changing. You get it? So definition is what? Is the process where two or more related species become more different over time, often due to different environmental pressures or different environmental habitats. Okay, now they share a common ancestor. So these two things are really important. Different habitats they are going, but they had a common ancestor. Species share a common ancestor but adapt to different environments. Example, 4 limbs of vertebrates. So 4 limbs of human beings, 4 limbs of bat, 4 limbs of whale. Everything, it has evolved differently, my dear children. Human beings have a hand, okay, bat have a wing, whale have a flipper, but if you see the structure of it, they have the same thing like us, radius, ulna, carpus, metacarpals and everything, phalanges, everything is there in them also, but they have different structures of it. But internally if you see, bones are the same, they have arranged themselves in different manner, giving rise to hand and wing and a flipper. So 4 limb of a vertebrate is an example. All of them basically they have evolved from a common ancestor. They are all mammals, my dear children. They have evolved from a common ancestor. So this is the thing. Okay. Convergent divergent evolution comes again. So obviously something that you need to know. Now explain the role of fungi as biofertilizers. What do you mean by fertilizers? First thing, fertilizers are something that increase the nutrient quality of the soil. Biofertilizers means they are basically organisms. They are not chemicals. They are organisms which are going to increase the nutrient quality. How? Probably they are going to fix nitrogen, or they are going to do something. They are going to have some add up in the soil which makes it really good. Okay, so explain the role of fungi as biofertilizers. How do you think that fungus could help in increasing the nutrient quality of the soil? First thing, biofertilizers are natural fertilizers that contain living microorganisms which enhance the soil fertility and promotes plant growth, supplying essential nutrients to the plants. Okay, fungi form symbiotic association, my dear children, with whom? With the roots of the plant known as mycorrhiza. The fungi help the plant by increasing the surface area for absorption of water and minerals. So what does this fungi do? They have hyphae, they move around the soil. Okay, they get into the soil, they have more surface area now for absorption of water and the minerals, especially what, my dear children? Phosphorus, which improves the plant growth. In return, the plant provides the fungi with organic nutrients like carbohydrates. So the plants are the one that basically gives what? They do photosynthesis and they produce what? Carbohydrates. So these organic nutrients can be given to the fungi, but the fungi instead will be taking and sucking in what? The phosphorus and things like that. Okay, similarly you have rhizobium. Rhizobium fixes what? Nitrogen, my dear children. So you have for different things, you have different organisms, but anyway you are asked about fungi. So yes, ठीक है, works. Chalo. Now, mention the evil quadrant of biodiversity loss. Again, something always repeated. Evil quadrant, as in you are asked about 4 things. Quadrant as in 4 things, okay? Which is responsible for biodiversity loss? Every single time this question comes, every single time you have to remember it, okay? So, the first is habitat loss of fragmentation. Because of this, obviously, you will have biodiversity loss. Over-exploitation of things, okay? You are overkilling organisms, my dear children, obviously they will be dying. Okay, they will be soon getting extinct also. There is a loss of biodiversity. For example, stellar sea cow. Example is stellar sea cow. Okay, invasive species, there are species which we have taken from outside because of its beauty and things like that, but when they came in, they are going to kill the organisms which are over here. For example, Ecornia, okay, or what you call as water hyacinth, it came in, okay, it was not allowing the other plants which are there inside the water to receive enough amount of light for photosynthesis, or probably it was depleting other organisms of oxygen and things like that. So they are doing a lot of things. These are invasive species. They were not from India. We brought it. Now, co-extinction, when one organism is dying, other organism which is dependent on it will also be dying. Okay, so these are the reasons for biodiversity loss, and this is the evil quadrant. Please go through it really well. Cool. Now, it's not like I am asking you to read it now only. I understand I am going really fast. I want you to read it later on. Okay, later on also you can read it. This PDF will be available to you. Just put down the comment that we want the PDF. I will be giving you the PDF. Okay, chalo. Section C consists of 3-marker question, my dear children. You have to answer any 5 of the following questions. Okay. Now, mention the different stages in dicot embryo development. So, in the picture also I mentioned you, this picture is very important, the most important diagrams. Okay. So, listen to me carefully. Dicot embryo, monocot embryo, everywhere. You have the embryo first known as pro-embryo. Okay, the pro-embryo basically it goes to globular stage where it's like circular stage, and then there's a heart-shaped stage, and then there is a mature embryo. So these are the different stages of what? Development in a dicot embryo. Okay, will you be writing these points? Yes. So these are the different stages in dicot embryo, and then all the points. Cool, and you get 3 marks. So, define the following: spermiation, spermatogenesis, spermiogenesis. Spermiation, what do you mean by that? Basically, the process where the sperms, the spermatozoa which are formed in the seminiferous tubules, they're finally going to move out. This is known as spermiation. Spermatogenesis. Spermatogenesis means the formation of sperm. Okay, from the spermatogonia, okay, the formation of sperm is known as spermatogenesis. Spermiogenesis. So basically, when the meiosis happens, okay, the primary oocyte will be dividing into primary spermatocyte will be dividing into secondary spermatocyte. Secondary spermatocyte will be dividing into 2 over here, 2 over here, 2 spermatids. So I call them kids. They are not matured. So spermatids have to again attain maturity, and this is known as the process of conversion of spermatids to spermatozoa is known as spermiogenesis. Okay, this is known as spermiogenesis. So spermiation is the process of release of mature spermatozoa from the seminiferous tubules to the lumen of the tubules. Now spermatogenesis is the process of formation of male gametes, spermatozoa, from spermatogonia in the seminiferous tubules of the testis. It involves mitosis, meiosis and differentiation. Spermiogenesis, the transformation of haploid spermatids into mature motile spermatozoa. So spermatids are the goal things. But do we have round things? No, my dear children. Sperms are having elongated tail. So they have to form the tail and everything. This process is known as, basically the conversion into spermatozoa is known as spermiation. Okay, so this is what I wanted to tell. I think all the teachers will be really proud of me for teaching you in this fast pace, because I am usually scrutinized for taking a lot of your time. But yes, this is for you guys only, that I explain way too much, because I think you might not be understanding, or I go from the basics because I want you to understand things better. But since we have already completed all the basic thing, there's no more basics over here. You have to go and watch the video and come over here. This is probably the 100th time that I'm telling you, but please understand the importance then. Now, what are contraceptives? Mention any 4 qualities of ideal contraceptive. So, contraceptive is a method by which pregnancy can be prevented. Okay, what? Pregnancy can be prevented. Mention 4 qualities of ideal contraceptive. Okay, should it come in between your sexual drive? No. Should it be user friendly? Yes. Should it be highly effective? Obviously, if you're using it, you should be believing it completely, right? Should it be highly effective? Okay. Does it have side effect? No. So these other things. Contraceptive are methods of devices to prevent pregnancy by interfering with fertilization, implantation or ovulation. Qualities: user friendly, highly effective, reversible and minimal side effects, my dear children. Cool. Chalo. Done. Okay. Now sketch and label Miller's experiment. This is the Miller's experiment where you have to label everything over here. What all things he included: methane, ammonia, water vapor, hydrogen gas, 2 electrodes, my dear children, in a chamber like this where there is high temperature. Okay, where there is high temperature, 800 degrees Celsius, okay, and also you have conditions like this where you just have these gases, water vapor and everything, and then what do you get from these inorganic things? You get organic content right over here, my dear children. So with this you get to know the chemical evolution of earth. So in the primitive earth there was only these gases. With these gases, how did organic content form? This is how organic content form, and this was the experiment of Stanley Miller. Okay, so you have to draw this and explain. Cool. Now with reference on, also please remember there is no oxygen over here. Okay, there is no oxygen over here. It is totally anaerobic condition. Now with reference to pneumonia, write the scientific name of causative pathogen, mode of transmission and 2 symptoms. Pneumonia is scientific name of the causative pathogen. Okay, Haemophilus influenzae. Second is Streptococcus pneumoniae. Mode of transmission, how do you get pneumonia? I am sneezing. If I am having pneumonia from the droplets, if I giving you shake hand, you will also, you take the droplets and put it in the nose, mouth, whatever you want. Okay, so basically there are high chances that this particular pathogen which is there, okay, Haemophilus influenzae and Streptococcus pneumoniae, both are bacteria, they can infect you. Okay, now the 2 symptoms, fever, okay, fever, high fever, and then respiratory issues, my dear children, that is one symptom. Also the grave symptom, if everything increases way too much, you will have blue nail, and the lips and everything will get changed, the color will be greyish and things like that. Okay, so causative pathogens, Streptococcus pneumonia or Haemophilus influenzae. Mode of transmission through droplets from an infected person coughing or sneezing, or direct contact with the contaminated surfaces or respiratory secretions. Fever and chills is there, difficulty in breathing and persistent cough will also be there in the case of pneumonia. Okay, now name the microbes that produce citric acid, acetic acid and butyric acid. I told you 10,000 times to remember this. If you are someone who did not take my word, okay, into serious consideration, then you are going to lose your marks. How many marks? 3 marks. But I know you won't be losing. So yes, so citric acid, my dear children, is produced by Aspergillus niger, a fungus. Acetic acid by Acetobacter aceti. Butyric acid by Clostridium botulicum. So here is it. Okay, citric acid, Aspergillus niger, a fungus. Acetic acid, Acetobacter aceti, a bacterium. Butyric acid, Clostridium botulicum, a bacterium again. Good. Chalo. Next, what are ecological pyramids? Mention any 2 types of ecological pyramid. So ecological pyramid is where you are categorizing organism according to their trophic level, my dear children. You have multiple types of ecological pyramids. Now what is the use of ecological pyramids? They give you information about the individuals, number of individuals, the biomass, the energy flow and everything. Okay, now any 2 type of ecological pyramid, then there is pyramid of number where you are talking about the number of individuals in each trophic level, then there is pyramid of biomass where you are talking about biomass in each trophic level. Okay, so graphical representation showing the relationship between different trophic levels in an ecosystem based on energy, biomass or number of organisms. These pyramids have producer at the base and successive trophic levels stacked above. So write like this, okay, your points are right over here. So please use these points and write your answers. Now pyramid of numbers represent the number of individuals at each trophic level. Pyramid of biomass represent the total biomass at each trophic level. Cool. Section D, 5 marker. Okay. You have to answer any 4, but you have so many questions. Chalo. Flowering plants have developed devices to discourage self-pollination and encourage cross-pollination. Justify. So over here you have to talk about outbreeding devices. Okay, outbreeding devices. So that is the whole question. So self-incompatibility is one. Should we bring my hair down here? Yeah. So sorry again. Yeah, self-incompatibility is a thing where what happens. Okay. The pollen of the same flower would not be able to fertilize the ovule of the same flower. So genetically they have modified themselves. Okay, there's a problem in this genetic makeup, okay, such that they have become self-incompatible. Even though their pollen come and land, still the fertilization would not be happening. Second thing is unisexuality that is there in the flowers. For example, if only both male and female flowers is there, then there are high chances of them to fertilize each other. What if one flower just has a male part and one other flower has just the female part? It becomes a lot more easy for self-fertilization to not happen, okay, for cross-fertilization to happen as well. Now the stigma and the anthers mature at different times to prevent self-pollination. That is also there. Stigma receptivity and anther dehiscence and things like that, maturation might be at different times. So obviously they would not be able to do self-pollination. Now these methods, altogether what do you call them? You call them outbreeding devices, my dear children. They encourage cross-pollination, but but what's it discourages self-pollination, because self-pollination can cause inbreeding depression. Okay, it can cause inbreeding depression where they won't be producing fertile offsprings later on. Cool. So all your points should be there. Now sketch the diagrammatic sectional view of human female reproductive system. This is the view that you have to draw. If you are not drawing, you are losing 5 marks. So you know how important this particular diagram is. Apart from this, I have mentioned all the important diagrams of your biology PU2 exam in 2 videos, most important diagrams in Brahmastra. Go watch it. Okay. Now, observe the following dihybrid cross and answer the following questions given below. Write the different phenotypes in the progeny. So, here I have not mentioned the answer, I will tell you the answer itself. So, progenies which you will be getting is, okay, yellow and round. Okay. So, over here if you see, zoom. Capital Y, it's for yellow. Capital R is for round, okay, and here you have small y green and small r wrinkled. So over here round and yellow is basically a dominant trait and wrinkled and green will be what? A recessive trait. So what are different type of phenotypes in the progeny that you are going to get? Listen to me. Round and yellow is there. Okay, this thing I think I have to write it. Where is it? So we have round and yellow and what? Wrinkled and green. Cool. Now we have to make a mix and match. Round was with yellow, this time we will make it with green. Okay, and wrinkled was with green, this time we will make it with yellow. And tada, you have how many progenies? 4 type of phenotypes. Phenotypes means the physical appearance in the progeny. Now, how many recombinant type of progeny are present? 2 recombinations are there. This is there in the parents. So I'll say 2. Now, identify the recessive genotype in the progeny. The recessive genotype as in the one which is not found in a lot of organisms. For that, I have to tell you the number of organisms which have this. So 9 of them will be round and yellow, 3 of them will be round and green, 3 of them will be wrinkled and yellow, and 1 will be wrinkled and green. So the recessive one will be this. So identify the recessive genotype, my dear children. What is the genotype of this? If it is wrinkled and green, it is recessive, my dear children. It should be small r, small y, small y. Or according to your question, it should be small y, small r, because they have put green first and wrinkled second. Cool. And what is the phenotypic ratio? I already told you the phenotypic ratio 9:3:3:1. With this, you get 5 marks. So we were talking about this. So just replace this, okay? Yellow round and green round and yellow wrinkled and green wrinkled. With that visible the case. So you just have to make the new combination that is there and you just have to name it. Always remember the dominant one is going to be 9 and the recessive one is going to be 1. Cool? Chalo. Next. Ting. Describe the steps involved in DNA fingerprinting technique. You have to go ahead with all the steps. Isolation of the DNA. Okay, you have to isolate the DNA. Then you have to digest them using what? Restriction enzyme. That is, you have to cut them. Now, after that, separation of the DNA fragments. This cut fragments have to be separated using what? Gel electrophoresis. After gel electrophoresis, what do you have to do? You have to transfer all this thing into nylon membrane. Okay, and after transferring into nylon membrane, still you cannot see this. You have to hybridize them, okay, with the probe that you're looking. So DNA which is there already found in the blood of the criminal or things like that, from there you got the DNA. That particular probe is being radioactive, okay, it is labeled radioactively, and now that particular probe is being run over here. So wherever it goes and matches, it will be showing the radioactivity under UV radiation, my dear children. So detection of the DNA bands, the last one, but the bound DNA probes are detected using X-ray films or fluorescence to visualize unique pattern of DNA bands. Now only if the person is having this complementary DNA strand, it will go and band, otherwise it won't. So you won't be seeing any activity over there. If you see activity, that means that guy is the criminal. Okay, so this is how DNA fingerprinting technique is used in finding the criminals. Okay, so all the points, write it properly. I have made it into color in bold format so that you can remember it profoundly. Okay, now the next thing, organs cannot be transplanted from just anybody. Why do you think organs cannot be transplanted from anybody? Because, my dear children, everything in our body is coated by proteins. Now everyone has different proteins on their surfaces. Surfaces. Now for identifying these proteins, my dear children, you have other things in your body. There will be other proteins, these proteins and these proteins, that is the cell surface proteins, which you call as antigens. Okay, are there. Now to detect these antigens, what do you have? You have antibody. Suppose the antigens are okay, like for example, our own cells, okay, our own cells have proper proteins which would not be igniting an activity over there. But if it's from some foreign cell, it will have some different antigen, and our antibody will think we have not seen this antigen, obviously we have to react to it. So this is why, since our body feels that everything that comes from outside will be a foreign particle and it can cause a trouble, it goes and tries to kill it, which is why organs cannot be transplanted from just anybody. And here the major role actually is not played by the antibody and antigens, things like that, but there are a lot of other things where cell mediated immunity is being involved. Cell mediated immunity is being involved, my dear children. So on our cells we also have MHC particles, major histocompatibility complex. These are proteins again. So these major histocompatibility complex are very unique to each of us, and it is being sensed by the T lymphocytes which are there, which is responsible for the cell mediated immunity. So they are the ones. So that is a question that has popped up differently, okay, kind of differently, and you might have faced trouble, which is why I am telling you the answer. Okay, so all the cells of our body have MHC. Okay, we have unique MHC just like our fingerprints. Now if some other or from some other animal or physically from human being we get the organs transplanted, what happens is that they have their own MHC, and this will ignite, this will make the cell mediated immunity active. The T helper cells, T cytotoxic cells, everything will be active, and they cause a huge problem, and they cause something that is known as graft rejection. They reject the graft. Okay, this is known as graft rejection, my dear children. Now mention the clinical test to be carried out before transplantation. What is the test that you have to carry? Basically the blood group test is one such thing that you have to make sure. Second thing which is there is the tissue matching test. Okay, the tissues which are there should be matching to each other. They should not be igniting a lot of troubles. Tissue, as in, it is made up of cells, and cells have all these proteins around it. So you have to mention or see whether they are matching or not. Now why should organ transplanted individuals take immunosuppressants all their life? Why do you think they have to take immunosuppressants? Because, my dear children, what happens, as I said, the body feels that this is some or foreign particle. Now it will obviously ignite an immune response. Now if it is way too strong immune response, it will be a problem for us. So what do? You have to make your immune system a bit slow down, okay, bit cool down, which is why you take immunosuppressants. So this is the answer. The immune system of the recipient may recognize the transplanted organ as foreign and attempt to reject it. Cell mediated immunity play a major role over here. Mention the clinical test, blood group matching and tissue typing to match the proteins on the surface of the cells, which helps in reducing the risk of rejection. Why should organ transplanted individuals take immunosuppressants all their life? Immunosuppressants are required to suppress the immune system as it could prevent it from attacking the transplanted organ. Okay. Next thing, discuss the importance of transgenic animal. Again, a question straight directly from your NCERT. Okay, so normal physiology and development. For that we need transgenic to understand the normal physiology and development. So transgenic animals can be specifically designed to allow to understand how genes are regulated. The normal physiology of the genes and everything we share and understand with the help of them. We can do study on them. Now study of diseases. So we can take them as models for study of human diseases like cancer and things like that. We can do investigation with the help of new treatments and things. How do they react to these treatments? Biological products. So we can get biological products, for example, alpha-1 antitrypsin. Okay, this is a protein that is used to treat emphysema. This was produced by transgenic organisms. So for production of biological products. Now vaccine safety. Now you have to test. Obviously you cannot go and test on human beings. So you do go and test on what? Transgenic mice. Okay, you are going to test its safety, whether it will be safe for humans. Now that is also cruel. That is also cruel, this is also cruel, everything is cruel, but we need a solution for that. Now chemical safety testing, that is, for example, we make an organism highly sensitive to even little amount of chemicals, and then we give the chemicals and see how are they reacting. How are they reacting? If there is a very grave reaction, probably we should not be using that much amount of chemical in human body, or probably we could make very less, or probably we should not use this chemical itself. So this is how they are used. So these are the importance of transgenic animals. You have to write each and every point that I have mentioned over here. This is directly from your NCERT. Cool. Now mention the various defense mechanism developed in prey species to lessen the impact of predation. So defense mechanism, you are asked, you are not asked about animals or plants, so you have to tell about both. Okay. So in animals, some species of insects and frogs are cryptically colored to avoid what being detected by the predator. That is one defense mechanism. Some are poisonous. Okay. So they are not eaten by the predators. Some butterfly, for example, monarch butterfly is highly distasteful. It does not taste good at all. Why? Because of its presence of chemicals in its body. Okay. Now in the case of plants, you have in acacia and things like that, you have thorns, like cactus, they have thorns. Okay. Now many plants produce and store chemicals. which when taken by the predators can kill them, okay, probably can mess up with the digestion and feeding things. Okay, now Calotropis produces poisonous cardiac glycosides against herbivores. You see the example over here, Calotropis, which is there. Okay, this is the Calotropis plant which is there. You might have seen this. It produces what? Cardiac glycosides, which is going to kill the herbivores which are going to feed on it. Cool? Yes. Now section D, there is one more section part where you have to attempt just one 5-marker question, and this is for that. Okay. So given below is the representation of amino acid composition of relevant portion of beta chain of hemoglobin from an affected individual. Identify the genetic disorder. What is this genetic disorder? Where my dear children, this is the peptide being made. Over here, what you see is in the mRNA, it was GUG, which was coding for valine. It was not coding for valine. Basically what happened, some replacement happened, okay, some replacement happened and you got GUG, and this GUG is now coding for valine at the 6th position. What is this particular disease? Well, this particular disease is known as sickle cell anemia. Name the amino acid that is replaced by valine at the 6th position. What is replaced by valine at the 6th position? Okay, what was there initially? Glutamic acid, my dear children. Glutamic acid. It was replaced by valine at the 6th position. What is the difference in the RBC of the normal and this affected individual? So whenever glutamic acid is replaced by valine, you are changing an amino acid altogether. If you are changing an amino acid, obviously the structure of the protein. What is this protein coding for? This is for hemoglobin, my dear children, right? So this globin chain is going to get affected. So what happens is that the normal cell shape, which is discoid, okay, the RBC is discoid in nature, the cell shape is discoid. It will now appear like spindle. So it will turn to spindle shape because of this particular thing. Now, who are likely to suffer more from the defect, the males or the females, or both the males and the females equally? So this is not a sex-linked disease. This is what autosomal recessive disorder. Okay, this is an autosomal recessive disorder. For example, if there are 2 alleles, my dear children, both of them are affected, that's when you have the disease. It can happen in both males or in the females. For example, if the male is having this condition, you say male have sickle cell anemia. If the female have this condition, have them both the sickle, this like this, you call that female has sickle cell anemia. So suppose these 2 genes are there. So this is a male obviously who has sickle cell anemia. Now if the condition is like this, okay, this is the female who has what? Sickle cell anemia. That's all. So it is a recessive autosomal recessive disorder, my dear children. And always remember the GUG, instead of GUG it was GAG, which got converted to GUG. Okay, and this GUG, my dear children, this GUG is responsible for valine, not glutamic acid but valine. But over here GAG was responsible for glutamic acid. That's how you get the disease. Now what happens when the normal cell shape is being deteriorated? It is now sickle cell. It won't be able to carry more amount of oxygen. That is a problem. That is how they die. Okay. So how do you write? Sickle cell anemia is caused by mutation in the hemoglobin gene on chromosome number 11. It is inherited in an autosomal recessive gene manner, not X-linked. Males and females have the same risk of inheriting the sickle cell anemia if both the parents carry the gene. Both the parents are carrying the gene. For example, the condition is like this. HbA is the normal, HbS is the other allele. HbA is the normal, HbS is the other allele. This is one parent, this is another parent. Both of them are carriers of sickle cell anemia. It will be only there, this particular person will be affected only if the both alleles are same. It's a recessive condition, right? So now, suppose this gamete comes from the male and this gamete comes from the female. Now you have HbS, HbS. Whether it's a female or a male, the child will be having sickle cell anemia. Cool? Yes. Now, the process of copying genetic information from one strand of DNA is termed, DNA into RNA is termed as transcription. Okay, write the schematic represent structure of transcription unit. This is the transcription unit. You have to mention 3' to 5' end, 5' to 3' end. Okay, on the 5' end of the coding strand, this is the coding strand. This is a template strand. Template strand is the one that undergoes transcription. Okay, please remember it is the one that undergoes transcription. So your enzyme will come bind over here. It will be moving from this direction. Enzymes always, RNA polymerase moves from 5' to 3' end. So in response to this particular strand, that is template strand, it will be moving. But there is coding strand, we say the promoter region is present on the 5' end of the coding strand because we have to give some reference to coding strand, right? Similarly, the terminator region is there on the 3' end of the coding strand. That's how you say. Okay, this is a structural gene which is going to be transcribed. Now, if chalo, if 5-AGCT 3' is a template DNA strand, write the sequence of mRNA strand with polarity. What will be the mRNA strand, my dear children? This is 5' to 3' direction, right? Now, we should be starting from 5' over here. What will the polarity? So, the new sequence of mRNA strand with polarity will be what? T. If this is T, my dear children, it will be A over here. If it's C, it will be G. If it's G, it will be C. Here, if it is A, this time it won't be binding with T. Instead, it will be binding with uracil. Why? Because it is an RNA that is being formed. This is a DNA. In the RNA, there is no thymine, there is uracil. Now, instead of T will still bind with A, C will bind with G, G with C. What about A? A will bind with U, my dear children, again. And this will be 3'. This is the polarity. This is how you have to write it. 3' to 5', and this is the thing. Okay. Now, both the strands of DNA are not copied during transcription. Why do you think both the strands are not going to be get transcribed? Listen to me carefully. If both of them are going to get transcribed, okay, they are going to produce two different type of proteins. Same DNA strand, they are going to produce two different type of proteins, because obviously, okay, this coded for this, the other guy will code for another thing. Okay, in each of them, if they go for transcription, there will be two different things, and it will be huge problem. So, if both the strands are transcribed, it will result in production of two different mRNA molecules, which would not only waste cellular resources but also potentially produce contradictory proteins from same DNA sequence, complicating gene expression regulation. So, it will complicate the whole process, which is why we do not want that to happen. Okay. Now, what is the use of gel electrophoresis in genetic engineering? What is the use of gel electrophoresis, my dear children? Gel electrophoresis is to separate DNA fragments, okay, according to its charge and also according to its length, okay, or basically size, not length, I will say size. Now, what is the principle behind separation of DNA fragments in the gel slab? The principle is simple. DNA is what? Negatively charged. The DNA is negatively charged. Now, suppose the cathode is over here, cathode is also negatively charged, anode is over here, anode is what? Positively charged. DNA being negatively charged, they will be moving away from the anode, cathode, because negative, obviously they will not stay together. They will repel from each other and it will be moving in the other direction, my dear children. Okay, and apart from that, the smaller fragments which are there, which are smaller in size, they will also be moving really fast. Now, the enzyme stain used for staining the DNA. How do you stain the DNA? First thing, for example, you have separated the DNA. You take a nitrocellulose sheet, you put it up there, all the DNA molecules will be coming up here. Now, you are going to stain it to view it. What is the stain that is usually being used? Very important question, always asked. Ethidium bromide, my dear children. What do you call it? Ethidium bromide. Okay, and ethidium bromide will still not be visible. You have to put it under UV radiation. That's when it appears to be brightly orange color bands. Now, what is elution? Elution is where you know the DNA strands is there. I have to take it out from the gel. This process is known as elution, the separation of the DNA from the gel which is there. Cool. So, how do you write it? So, first thing, gel electrophoresis is used for what? It is used to separate and analyze DNA fragments based on the size. Gel electrophoresis, how does it work? What is the principle? It separates DNA fragments based on the size and the charge. When an electric field is applied, DNA fragments move through the gel with smaller fragments traveling faster and farther than the larger ones. The most common stain used for staining the DNA is ethidium bromide. What is elution? Elution is the process of removing or extracting DNA fragments from the gel after electrophoresis. That's all. Okay, and with that, we are completing all the questions of this particular paper. And for the first time, I completed a full length paper within 1 hour 4 minutes. Kudos to both of us. Okay, and with this, as I was saying, did you see any question which was very new to you apart from the organ transplantation? It is also not very new, but I know you might find it difficult to frame the answer all at once. So I hope I was able to do that. If you still think that I need to explain on that, you can put down the comment. I'll explain it to you there itself. Okay, now that is there. Apart from that, I am really happy that all the questions came like that. If you also feel the same, definitely let me know. All my efforts have been of use. That is something which is very much important for me, as a teacher. So kudos to you, to all the people who were there throughout this video, and wishing you all the best. For the people who are still left to write your exams will be really good. Stay tension free, just give your 100%, my dear children. Everything is going to be good. I pray for you daily, and that's all. Wishing you all good luck, and do not forget to like, share and subscribe to our little channel, that is Diksha Karnataka. Bye-bye. Take care.